it is extremely important to bring new tools and new knowledge or new ways of thinking into the lab on a regular basis in order for us to move forward into out of the comfort zone into new spaces that allow us to make discoveries.
Welcome, Nicolas. It's a great pleasure to host you here in Human Technopole. Thank you for being here and to answer to our curiosities. So your lab was the first one to create blastoids, which are in vitro models that recapitulate early embryonic development, specifically the blastocyst stage, right? So can you explain what a blastocyst is and how is it recapitulated in the blastoid model?
Yeah, thanks. Thanks for inviting me first. I'm very happy to be here. And so the blastocyst is the very early mammalian embryo during the first week. And the particularity is that, like many species, we float at the beginning, but we mammalians are implanting into the uterus. And this enables to develop an interface with the mother that is very specific and that has allowed us to evolve into what we are. And the goal of the blastocyst is to implant. So that's where the principles of the blastocyst come from. And of course, studying all this is extremely complicated because in mammalians, first, we don't make a lot of embryos, and those embryos are in the womb. So it is somehow important to develop a stem cell-based system that could recapitulate all this in a dish in order to study what was previously inaccessible.
And on this topic, how do blastoids help us understand the very early steps of human life? In which way?
Well, they allow to study those processes of early specification and early morphogenesis, early patterning. So all these are classical dev bio questions, and we studied them extensively with the real thing, the blastocyst. But by having blastoids, we can do screens because we can generate them in very large numbers. We can do genetic engineering by changing one cell type and not the other to understand the interactions that are happening. So we can do things at a much higher scale and precision. And of course, this allows us to, in the case of the mouse blastoid that we published in 2018, it allowed us to transfer them back into a uterus. And of course, because it's complete and because it's pre-implantation stage, it is capable of implanting and eventually to develop into a full organism. Nobody has done it yet, and there's a lot of progress on that field. But and I think that this will allow to study all the subsequent stages. In the case of the human, of course, there are ethical limitations here, but we can combine them with endometrial organoids to mimic this process of implantation in a dish.
Cool. So we could describe embryonic development as a process of self-organization. It happened that you used the analogy to birds flocking. So how do you think blastoid can help us to understand how the cells self-organize in the embryo?
Yeah. Well, so there are many different things to study in biology, and historically, in the last 75 years, there's been a lot of gene-centered research because we discovered the genes, the regulation, and of course this was all very interesting. But there are also other ways by which our organisms are actually developing, and those other ways are actually not new. We know it for a long time. For example, Alan Turing, the famous mathematician, proposed some models for patterning of, for example, the skin of the cat with the different colors. These are not happening. These are happening via self-organization, meaning that the cells are cross-talking with positive and negative feedback loops, and all this self-organizes in order to segregate, in that case, the colors on the cat skin. So like those type of principles have been historically understudied, and so there's a lot of interesting stuff that can be discovered based on those ideas that are actually quite old.
Cool. I heard you also mention implantation, which is known to be one of the most fragile steps during pregnancy. And so we were wondering how these blastoids can help the fertility treatments and potentially also prevent early pregnancy loss.
So this is something that I'm extremely interested in, because of course we are doing basic science in the lab, but we also set up a biotech in order to do the whole translational, and they are using blastoids in order to screen for molecules. And they already have found like a handful of molecules that are now moving into the clinics. So we can find molecules that are going to improve the formation of blastocysts in a dish in the IVF clinic. And this is what we are currently doing. But what's also extremely interesting is that we humans are massively failing during the first weeks of pregnancy. There's about like 50 to 70% of the embryos that are just lost. And we don't know why. And we barely know that we are pregnant at this time because it's extremely early. And so we need to, but this happens at the time of implantation. So now we are in the capacity of understanding those things that are are totally inaccessible until now. And there are a couple of theories because actually this embryo loss might not be totally unpurposed. It could be evolutionary tinkering where things are going wrong, but it's okay because we still make babies. But it could be also that there are some conflicts that are happening, genetic conflicts or chromosomal conflicts that are happening that somehow select for specific pregnancies or specific embryos. So these are the type of questions that I think are extremely interesting and can be tackled in the next decade.
That's very interesting and promising also, thinking ahead. Instead, I wanted to bring the discussion more on the ethics side. So we know that human embryo model can raise important ethical questions. So can you explain to the public the difference between a human embryo and the blastoid model and how this distinction is relevant for science and society?
So I think like we as scientists, we are like the first one to actually come across those potential ethical questions, and we are the first one who actually like bringing them up. So we see things happening in the lab, and we're like, okay, we need to engage with ethicists, philosophers, and this is what we did since 2018. We are working hand in hand with different groups of ethicists, philosophers, lawmakers, in order to make sure that we are providing solutions to ethical problems, and we are also building frameworks so that the research is done in extremely good and justified way. And so this, what I truly believe is that these type of models are of course extremely different from embryos. They are not. We know that they don't have the capacity of forming an organism, but that they are solving an ethical challenge. So we now can do research that we could not do previously because of ethical restrictions. So these are more like solutions than problems.
This field requires the background, let's say, from many different scientists, from physicists to biologists to engineers. And so we're wondering how crucial it is, this interdisciplinary and international collaboration to bring up such a project and to understand the early stages of human development.
Yeah, it's super interdisciplinary. I'm an engineer by training, and then did and every we are changing a lot, and we are integrating a lot of different things. Now we are working a lot on evolution and gene regulation. So it's extremely important and interesting for as a scientist to actually bring new type of knowledge into your research. So we are doing this on a regular basis, kind of like retooling the lab and also like allowing us to like think in different ways. And this is the way that you actually make discoveries, like by not staying in your comfort zone, but just like like trying to bring new people with like like very different ways of thinking. Yes. That's the very interesting part in fact.
Having different points of views. Yes. I
just
agree.
bringing people who are thinking differently.
Yes. Well,
thanks.
Thank you so much. We look forward to your seminar
Thanks so much.
this afternoon, and I hope you'll enjoy your stay here in HD.
Yeah, sure.